Okay, here we are going to do a quick review of cellular respiration. And my slides are not great on this because you guys made your trifold, which was the most important part of this, right? So again, photosynthesis, building up of glucose. We are really focusing here on cellular respiration, which is the breakdown of glucose to form ATP. Remember, sun is our main source of energy on this planet. It is used by the autotrophs to build up organic molecules in the form of glucose, which then mitochondria can break down, right? So cellular respiration, we are really using chemical energy stored in glucose to regenerate our ATP. Whereas photosynthesis, you are making glucose with the presence of oxygen and light energy. So cellular respiration is the catabolic pathways of both aerobic and aerobic respiration, which break down organic molecules to make ATP, right? So there are kind of two parts of this. You can have fermentation and anaerobic respiration, requiring no oxygen, occurring in the cytoplasm, releasing a little energy, or you can have aerobic respiration, meaning you are using oxygen, it's going into the mitochondria, and you're releasing a huge amount of ATP. Cellular respiration is a series of redox reactions. Remember, redox is oxidation reduction reactions, where oxidation is a decrease in energy, and we're really looking for a loss of an electron or a loss of a hydrogen. In reduction reactions, you are gaining an electron, and you are gaining an H, and you are increasing in energy, all right? So here, when we look at the reaction of cellular respiration, glucose here is losing hydrogens and electrons to become oxidized into carbon dioxide. Oxygen is gaining electrons and becoming water, right? And that's a reduction. Okay, there are many coenzymes which are important for the breakdown of glucose. In particular, we are talking about NADH and FADH2. These are high energy electron carriers, right? We are going to accept these electrons and carry them, kind of carrying the energy within a cell. That's all I'm going to say about those. So what we're going to see in cellular respiration is that we are going to kind of move energy through an electron transport chain to generate a lot of ATP. We have three main phases. The first phase is glycolysis, where we are going to break down glucose into pyruvate, right? This happens in two phases. Glycolysis has two phases: an energy investment phase, where you're inputting some ATP, and an energy payoff phase, where you are gaining some energy out, right? But the whole purpose, glycolysis, break down glucose, you make 2,3 carbon pyruvates, right? Glycolysis is the most ancient of these processes, and it occurs in the cytoplasm, right? If you have oxygen around, this 3 carbon pyruvates can go into the mitochondria through the Krebs cycle, right? We also talked about before the Krebs cycle that pyruvate has to get converted to acetyl-CoA through pyruvate oxidation. And then it will go into the Krebs cycle where we get a lot of carbon dioxide generation. In addition, we get some of these high energy electron carriers in the form of NADH and FADH2. The electron transport chain and chemiosmosis together make oxidative phosphorylation. I want you to think about that term, oxidative phosphorylation. So what's being oxidized here? What's being oxidized are these high energy electron carriers. NADH and FADH2 get converted to NAD or NAD plus or FADH, right? And that is when they go, they get handed off to the protons on this inner membrane. Workout. And then through this oxidative phosphorylation, that is where, and the phosphorylation is when ADP gets made into ATP through the process of chemiosmosis. So most of the energy that we make in ATP is through this oxidative phosphorylation, meaning the electron transport chains, and chemiosmosis with ATP synthase. However, if we have glycolysis and the citric acid cycle or the Krebs cycle, we do make a little ATP there, but that is made through substrate level phosphorylation. And that is just when an enzyme literally takes a phosphate from one thing and sticks it onto ADP to make ATP. So you should look at your trifold, go through all those steps again, ask me if you have any questions. I'm not going to get crazy on the bookkeeping of how many electrons and how many ATPs and ADPs, etc. You should know the flow of things. You should know the ins and outs of each. However, when we talk, I'm going to talk a little bit more here about oxidative phosphorylation. So here, the electron, whenever you look at a diagram, orient yourself. You got to figure out what's the inside, what's the outside, where are things flowing, what are the axes, etc. So here, the beginning is here, which this light yellow here is going to be the mitochondrial matrix, where NADH and FADH2 are being oxidized when they enter the electron transport chains. When they get moved along this chain, hydrogens are pumping into the electron, they're using the energy of these electron flow to pump into the intermembrane space. That energy in the electrochemical gradient, right? It's an electrical chemical gradient. Chemical because there's H's, electrical because they're positively charged. That energy in the electrochemical gradient will then be utilized to create ATP by ATP synthase. As these H's flow down their concentration gradient, ATP synthase turns like a turbine and creates ATP. This is oxidative phosphorylation, which is different than that substrate level phosphorylation. You generate a lot of ATP with this. So about 38 ATP made for every single glucose molecule, right? And you can figure out where these are coming from. But in general, glycolysis will give you a little ATP, a little NADH. Pyruvate oxidation will give you NADH. Krebs cycle, a little ATP, a lot of NADH and FADH2. Those will feed into the electron transport chain where you get a lot of ATP out, right? If there is no oxygen present, what happens? So if you have oxygen, this pyruvate will go straight into the mitochondria for aerobic respiration. If you do not have oxygen, then your cells will go through what is called fermentation, right? Which requires no oxygen. It occurs in the cytoplasm and it releases small amounts of energy, always through glycolysis, right? Pyruvate here from glycolysis, if there's no ATP, it will go through fermentation. Two different kinds of fermentation. If you are a yeast cell, you will go through what's called ethanol or alcoholic fermentation. You are going to make CO2 as waste there. If you are human, you will go through what is called lactic acid fermentation. You will make lactic acid as a byproduct. The most important thing about fermentation is that it occurs when there's no oxygen, and the whole point is to regenerate your NAD+ so that you can do glycolysis again, because glycolysis does allow you to make a little ATP. Realize that different types of food in your diet feed into this process of cellular respiration in different parts. And cellular respiration is controlled by different inhibitors, allosteric inhibitors and allosteric activators. Remember, allosteric means we are binding away from the active site, meaning we're dealing with enzymes. The most The best example here is the enzyme called phosphofructokinase in the early steps of glycolysis. It can be activated by an allosteric activator called AMP, meaning we don't have enough ATP around. I'm going to stimulate this so we can run this faster. We can also, if there's enough ATP generated through this whole process, and we want to turn this system off, ATP itself acts as an allosteric inhibitor of phosphofructokinase, saying glycolysis, stop, we don't need any more ATP. All right, and that is a very quick cellular respiration.